Anyways, so I would say maybe we just begin. My name is Marina Lova. I'm from Biotech. And in today's session, I would like to speak with you about the challenges when you work with the membrane proteins and about our solutions with a focus on the automated workflows. And today in my talk, also a colleague from Jena Analytics is going to have a couple of, also a part of the talk together, because this is our collaboration partner, which helps us to basically implement our solution to our end customers. So just a couple of words about the Biotech. We are based in Germany, in the city Monheim, so it's near Cologne. But we also have a production site in Göttingen, as well as a distribution facility in the US, which, of course, makes the life of our US clients easily. We were founded in 2012, and we are working basically worldwide, helping people when they have challenges with the membrane proteins. So when you work with the Biotech, we have different solutions on how we can help on your research. So on one side, we have products which help you to stabilize and purify proteins. It should not be specially in our membrane proteins. It's also applicable for the soluble proteins. For those who would like to outsource the activities, or targets may be also very challenging in the moment, so we have also services, so basically from gene to protein and gene to structure workflows. And a special path as well. We have a membrane protein catalog with human membrane protein targets. Basically, they are as ready-to-use projects, so you just basically need to tell us the target, the amount, and your downstream application so that we can find the best solution. So as you see, we have a main focus on the membrane proteins, and why is it actually important? So basically, up to 75% of the top-selling drugs on the market target membrane proteins. And this is basically this big fraction, of course, of the targets. So that's the most challenging targets, because, so we're going to speak with this anyway in a couple of slides, but you need to consider a couple of things when you work with the membrane proteins, especially. Of course, membrane proteins are surrounded by the bilayer, so they are incorporated in the bilayer mostly, and if you would like to purify it, you oftenly would remove it. And once you remove the bilayer, and the protein is exposed to the watery phase, so it just starts simply to aggregate, because it has basically part which is lipophilic, and the part which is or hydrophobic, and part which is basically hydrophilic, so that's why the whole structure collapses once you don't have the bilayer. So what do the people do with this? The classical way would be go with detergents, so you just basically wash away the lipids. You can also do thermostabilized proteins, so where you do the mutations basically, or you can cut even certain parts, so for example extracellular domains, and work with those. But the problem is, the question is here, is it the same protein in the end? No? So that's why people started to reintroduce the bilayers, for example, in this solution with a MSP nanodisc, so where you have a protein scaffold, so and you introduce the lipids back. So it helps partially, of course, But if you look at the biology, of course, is always way complex than we think about it. So we have in the bilayer not only 1, 2, or even 3 different lipids. We have a whole moiety of functionalized lipids as well. And how to reestablish this basically, so you cannot go for scratch. Even if you do lipid extracts, so that can be a challenge, because how we are sure that your composition of the lipids is in that moment native? No, that's not native anymore. So what people do, as I said, classically, so you would go with a detergent. So basically you would disrupt the bilayer. You would stabilize the membrane protein in detergent micelle, because detergent is an amphipathic molecule as well, so it would stabilize the hydrophobic domain of the protein. But you have different problems with this. And the first problem would be, of course, in the terms of the purity and yield. So there are membrane proteins that are simply not solubilizable or with detergents, or they're prone to aggregate once you start doing this, yes, or they're not stable. The other part is, of course, the purity. No, you need to definitely, so in this example, you have elution fraction after the affinity purification, so you see it would be necessary in certain conditions, yeah, so for sure to optimize the processes. And then when you go downstream applications, yeah, this is a DLS data. Of course, here you have still detergent micelles, no, and that's something that interferes with your downstream application as well. So it's sometimes hard just to use those preparations, so in the downstream. And of course, the third problem is that temperature, so the stability of the protein is compromised, basically. So most of the targets start to unfold. So the melting point would be like starting around 40 degrees. Yeah. So and basically that means for you that the protein, if you want to measure it for a longer time, it will probably not give you reliable results in the end. So what's the solution? How we can deal with this? Here at Biotech, we use synthetic copolymers that stabilize membrane proteins as native as possible. So how does it work? You will basically take a bilayer, or we take whole cells, we take cell lysates, we can extract membrane, so it doesn't matter which kind of membrane it basically is. And we throw the synthetic copolymers, and they start to interact with the lipids, and they wrap around the protein, and you can imagine it's like a cookie cutter. So it's basically takes the protein out of the membrane as it is, together with the host cell lipids. And this is the difference. So we don't disrupt the protein in this moment. And then you have this kind of the nanodisc, which is very, very stable, and you can basically handle your protein as it would be the soluble one. It's compatible with any physiological buffer. It doesn't need any additives, so you can go ahead and have your protein. If we look at the comparison between the protein stabilized in the detergent and protein stabilized in such a synthetic nanodisc, we directly see here the comparison. The proteins come mostly very pure, directly, without any optimization. You can see on the DLS, of course, we have no any additions in the buffer, so that means that you can really nicely and clearly measure the proteins. And what's important, your stability of the protein increases. Why? Because you have lipids, because the lipids interaction do matter. So as a summary of this technology, so basically we take out the protein together with all the lipids. So that's why if you compare different stages of membrane protein preparation, we work as native as possible. And the good thing about this technology that it works with every single membrane protein class. How is it possible? Very easy. We have a huge portfolio of synthetic copolymers. So basically this portfolio is based on the 5 backbones. Probably you already heard about SMA and DIBMA. These are older copolymers, so making those small particles. So people sometimes hear about this. But there are a bit older ones, yeah? So we made a huge step forward and made our own copolymers as well, which we call next generation copolymers, yeah, so which solubilization efficiency is way, way better. And from all of these 5 backbones, inside we have more than 30 different copolymers where we can basically screen and definitely find the one that would work for your protein. Mostly it's not only the one, so that's why we can take a look then which one would be more suitable for your downstream application. So what can you do? Basically anything you can imagine. So you can go for example, screenings of the bindings, for the high-throughput screenings, for the binding validations, so using the state-of-the-art techniques like MST, SPR, BLI, GCI. You can go into biologic developments. It works in vivo and in vitro. We have also a couple of examples here on the side. You can go for structural determination using cryo-EM or X-ray crystallography. And of course, you can go for the small molecule drug development using techniques like ASMS and DEL screens. So I would like to show you a couple of examples on how it looks like. So let's go for the example of the structure. So I have here an example. This is our, let's say, working horse protein. P2X4, it's a pain receptor, it's a trimer, and it's highly glycosylated, so and basically also stabilized in this NativeMP nanodisk. You get structure with a very, very nice resolution. Yes, so here in example 3.1 angstrom, you see all of the resolution of the post-translational modifications, and you also see, this kind of native transmembrane region, which is incorporated in the lipid bilayer and not somehow disrupted by the detergent in this moment. So here again, so that's something what our customers like to do in the service, for example. They come with the target and they say, okay, we have a couple of small molecules candidates, yeah, so we would like to see the binding. So this is a good preparation, to make those screens, for example, to see where the molecule is binding, for example, because resolution is that great. And that works, of course, not with only one protein, but it works with every single protein. It's only the question to find, the right combination of your target and of the synthetic copolymer. So basically, the screening is the heart of the whole work using our NativeMP system. Let's take a look on the small molecule drug development using ASMS. So we went into the collaboration with Momentum Biotechnologies, and they performed, this is the workflow from them. So basically what they do, they mix our protein with a library of small molecule compounds, and then they extract the fraction which has actually bound to the protein and analyze the hits. So we did an experiment where we so basically, where the Momentum measured 50,000 small molecule compound library, and the target was the GLP-1R receptor. So this nice target was very hot target already last year. So if you ever heard about Ozempic or Wegovy, yeah, so that's basically the target of this drug formulation. So in this screen of 50,000 compounds, we found 75 primary hits, which is a very good result for a GPCR, and 34 were confirmed hits. So they were confirmed against an empty nanodisk, so we are also capable not only to catch the protein into nanodisk, but also actually make an empty nanodisk with the lipids that we use as contrast screen. So that would be the same lipid combination basically as in your host cell in this moment, and ending with a 45% confirmation rate, which is great. We go even further and we take a look on different GPCRs, so that's what we called hot diabetic targets in this moment, so GLP-1R, GIPR, and GCGR, and even we can find compounds that show high selectivity, for example here against GCGR, which is not binding, so here down you can see, and which selectively bind GLP-1R and GIPR. So, but that's ongoing research right now. We also had the collaboration with XChem, where we have shown a kind of delability screen. Yeah, so that's the technology is also capable of, on using the DEL basically, DEL screens. So here also, because we also produce our own products, yeah, so we were able to use our own magnetic beads, yeah, so for the workflow, and basically it was shown that different protein targets stabilized on different copolymers were all basically capable of performing this kind of the screening. What's of course also possible, and that's why we love NativeMP system, is that it's capable not only to catch a single protein in the membrane, but actually extract whole complexes, because the copolymers, they basically adapt to the size of the protein. And in this example, you see our prominent protein, acetylcholine receptor, which is very, very hard to produce recombinantly. So that's why we basically extracted from the natural source, from the Torpedo californica tissues. We have even made our own resin with neurotoxin that selectively binds the acetylcholine receptor. And in this way, we can extract the native protein from the tissue, in this case, a big complex. So when the NativeMP platform is that powerful, how we can do this even more helpful for our customers? And in some certain moment, we came to the solution of the automation, because of course, something what is always asked is that when you screen your proteins and screen your conditions, you want to achieve, of course, high reproducibility. You want to reduce errors because you work mostly in the first pre-screens with very small volumes, just to see what's the output in the end. Yeah. So, and of course, yeah, I said there was like more than 30 copolymers, so doing it by hand takes time. Yeah. And usually, yeah, as you saw already in workflow with detergent, yeah, so it takes at least 2 days to purify such a protein on this kind of the screen, yeah, so it also takes several days. So once we started to establish our membrane protein catalog, we came exactly into this problem, yeah, and the problem of scalability, yeah, so on how you can do a lot of targets in their short time. And that's at the moment where we started to think about the automatization, and that's what came out. This is our plate, basically the optimization plate. And it consists of several rows, so to say, or lines here, which basically do the same job as scientists would do in the wet lab at this moment. So basically what you do, I will show you the workflow in several slides. But basically the idea is that you have synthetic copolymers. So here is an example with the cubipoles. You have a row with the magnetic beads, and that can be basically any magnetic bead, yeah, so possible. And once you proceed further in this plate, yeah, so you end up in the empty vial here where the protein eluate comes out. So basically, as it was developed, we were calling it lab on the plate. So something that you just insert, and in the end you get the protein. So this is the perfect imagination of this, and it's worked. So here's a bit more. So we have a special technology on how to prepare those megbit films. Yeah, so that's a bit different as in different vendors. And in terms of the magnetic beads, so we have very different affinity flavors. So we can go, if you work with a streptac, so we have a whole palette with a streptactine, XT, streptactine, or streptavidine. If you work with the histax, we have nickel NTA, the standard one, so, but also nickel indigo. This is the patented solution of Biotech, where you basically can work with the elevated amounts of ADTA and DDT without, maybe this problem that the columns turn brown, yeah, so when you work with them. So that's something indigo, yeah, has an advantage. GST tags, or our working host, the Rho1D4 tag, that's basically the same as the FLAG tag, but it's less charged, so it works perfect for us in terms of purification. So how's the workflow with the plate look like? Very easy. So you need to, first of all, I always say, if we can overexpress the protein, if we can get the expression, we can get the solubilization. So the first important point is, of course, to overexpress your membrane protein and to confirm, okay, we have a positive Western blot signal. Then you spin down your cells to the cell pellets, and then you open the cells. So we usually sonicate them, yes, but in general, it's also possible to use different cell opening techniques. So you spin down the cell debris, which is usually done by 9 kg for 30 minutes, and then you have this ready-to-use, so to say, cell lysate that is pipetted basically into the plate, and then you put your plate into the automatization machine, yeah, so, and press play. And in this moment, I would like to give a word to Andreas, yeah, so that he explains a bit what's actually happening afterward and how the machine works, yeah, so, and then we'll take a look on what comes out.
All right. Thank you, Marina, very much. Thank you all for being here. Thank you for having me in this chat. It's a great cooperation that we created here with Biotech. I'm Andreas. I work for Analytic Jena. And Analytic Jena is an instrument provider, obviously, for liquid handling and lab automation instrument, among others. We also produce other stuff, but that's not part of that talk. And so our expertise is not in the in-depth chemistry of doing things, but in translating things into automation. And when we have a look at the lab on the plate technology, we have several preconditions for automating it. So we need a robot that must have at least 8 channels pipetting, because as you can see, we're working from column to column to column, and each column has 8 welds. Then we need a robot that can transfer from a single tube to the plate, because that's usually how you would have your cells. You could also transfer from a deep well plate if you have them in a plate before, but in that workflow it was from a tube. The robot needs to pipe it up to 1,000 microliters, and it must be able to magnetize, because the proteins basically were entangled on a kind of magnetic beads, and these need to be cleaned up by the robot. Furthermore, it needs to be able to incubate, that means to shake and heat deep well plates, and transfer these deep well plates on deck. Because if you need them on the magnet and on the heater shaker, they need to be moved in a kind of way. And yeah, obviously these are like the technical conditions. Of course, there's also like ease of use conditions, like it must be small, budget friendly, things like this. And our answer to this is actually our working horse. It's a SciBioFelix, which is a small benchtop liquid handler that can pipe it from one channel up to 384 channels. And it can be tailored to the scientific applications like we did here with the Cube application. So first of all, starting with the housing of the instrument, so you can have like the SciBioFelix basic unit with an enclosure, as you can see it also on our booth at SLAS. But furthermore, there's an opaque enclosure for light-sensitive applications. There's a UV deck for UV deck decontamination. But furthermore, and this is very interesting for that group of customers because they also work with cells, you can actually have it without an enclosure. And then it's so small that it fits actually under a lab clean bench, so under a biosafety cabinet. The design is very compact and integration friendly in a way that you have 2 deck layers, and that actually results in a very small footprint. And so you have an upper deck, which is like split, as you can see in the animation. And we have a bottom deck, which is not splitting, but moving. And the decks are moving back and forth, and the pipetting is moving in the horizontal and vertical axis. And with this, like, we can actually address every kind of position to a very narrow extent. So it's even like we cannot only pipe it directly in the center of a well, we could also work with an offset or rinse slowly from outside, which is important for customers dealing with organoids. As mentioned, we have 12 positions on deck, a volume range from 1 to 1,000 microliters, and we can work with formats up to 5036 well. So don't get me wrong, we don't have a 5036 well head, but also the other heads, the 384 well head, can address 5036 well channels. But also, as you can see below, it's indicated rows, columns, single pipettes. And how is that working actually? So I included a picture of a 96 well head from below. The head has like 96 holes, and the tips are not sealed against the cone, like many other manufacturers do this, but against this kind of silicon mat, which you can see here. That's basically the mechanism. And we choose this mechanism because it's very robust, very service-friendly, and it provides high precision up to 1 to 2%, even in low volume ranges, or down to 1 and 2%. And now what you can do is you can either pick up a full robotip tray, then you would stamp 96 tabs at a time, or you can connect it, like you see these 4 holes here, with an 8-channel adapter. Then you can work with an 8-channel. Or you can also use this kind of adapter in combination with a tip box that has no frame to come from outside and just pick up 1, 2, 3, 7 tips, whatever you like. And that kind of combination, having the small instrument with that technology, really provides a lot of value also for this application. Other than that, there's also the idea that we can address 8 channels individually. This is a special kind of pipetting head where you could do a lot of hit picking or normalization application. Here you could do, for example, serial dilutions, normalizations, and hit picking. And as you can see in the animation, like each head is picking up a different volume. So this could also be available. So I would say I explained to this way right now, like a little bit about the instrument and that it can pipette in the channels. But what is more needed is also kind of the accessories, so that you can actually do tasks like magnetizing the beads and incubating it. And here you can work with a gripper, as mentioned, so you can transport plates from one location to another. You can work with a magnetic adapter, so you can also magnetize the beads to wash them or rinse them. Of course, there's also a waste box for tips that are left. Then there's a heating and shaking unit, which is a bioshake. And then there's also tube adapters so that you can work from single tubes and pipe it into the full plate. And also when you have high throughput applications, which are also a little bit planned for the future, small sneak preview, you can use a tip wash station. So you would not, you can even lower your budget cost, not reusing tips, and it's of course also more sustainable. Software-wise, you're really free to integrate every kind of protocol you want. So either you can use a composer, which is a software that allows you to freely script anything from aspiration speed, dispensing speed, locations, speed of the head movement, offsets, things like that. So if you don't want to pipe it directly in the center to not harm your organoid, you can do this. This is like a powerful tool, but requires a little bit of training. So then you can work with it and can script freely. You can also change your protocols later. Other than that, there's the Application Studio, which is an easy operation software for everyday users that just pre-select their protocol, which could be Cube, Plate, XMP, and then just press start and everything is starting directly. Yeah, so I would say finally we can say that that the robot is capable of all the features that is needed for this workflow. And we've actually shown this not just in internal experiments. So we have 2 app nodes. One of those you have here from the Cube app node. You could also have it digitally here with this QR code. And then there's also a second, more recent app node from a customer in Boston that we also did together with this customer, which also shows that not just the workflow can be done in 2 to 3 hours, but it also gives a dramatically increased yield, and it's just like more effective. Like you can see here from the manual workflows, we needed 132 grams of insect cells to get a yield of 180 micrograms. And here we can say, like with the SciBioFelix and the automated workflow, we needed 22 grams of cells for 70 micrograms of protein, which is a 4-fold yield increasement. We even had like 30-fold or 31-fold yield increasements. So it's actually really a game changer because it slows or it accelerates the process so much. You need like 3 hours now for something that you needed 2 to 3 days before, and you get a higher yield and a higher reproducibility. And yeah, I think that's a great co-achievement, and we have further data about this workflow that Marina can present in the next slides.
Yeah, thank you very much for your contribution, of course, for this great collaboration. And of course, it sounds now nice, yeah. So, but those are numbers. Let's see on how it really looks like on the gels and the data on how the protein looks like. So this is something what we get out of the instrument when we run out the plates. So you have here examples of the 3 proteins. So LAMP1, GLP-1R, and GIPR. So you can see basically the screen. So as I shown you already in the plate, so we would use here our cubipole backbones. Yeah. So and you can see that depending on the target, of course, it depends mostly on the expression level first. Yeah. So I mean, if you have like very good expressing protein, you would definitely see a band like this on the gel directly. So what we see here is basically that what comes out on the plate in the last while. So the elution fraction after affinity purification. So and this is protein which was not polished by the size exclusion chromatography yet, and it comes in this quality. So in this case, the LAMP1 was a very nice expressing one, and you can see that the performance of the all copolymers now they're similar. Yes, so in this case, so we have on GLP-1R and on the Gipper, so here upper band, yeah, so very remarkable results because those two are GPCRs, and GPCRs with detergents, it's a pain. You don't want to do this like this. So this would be kind of a standard workflow we do with the proteins. So we run our solubilization screen, which I said it's a heart of our technology, so that it's used the downstream application in the end. We take a look on the SDS page and Western blot, here in this case. And what we also do, we take a look on the DLS and nano-DSF data. So I have some DLS data here, and you can see among the copolymers tested, you see that the particle distribution is very near. So you have really homogeneous particle which coming out. And what's important here also, we take a look always something called polydispersity index, yeah, so that's a measure for the, like how homogeneous is the sample, yeah, so and usually what we say something near 0.2 and lower, it's a very good result in this moment, and you can see from the almost off copolymers, yeah, so we are exactly at either lower at exactly this threshold, which tells us that the quality is very nice. And because it's looking like this, we can directly go with those samples on the cryo-EM grids. And yes, without size exclusion. So usually that would be a horror story for all cryo-EM scientists. So they tell how you don't do the size exclusion. No, we can directly per screen, because I said we need to know which combination of the target and the copolymer is working. Yeah, so that's why usually what you do is you do a kind of grid screening in this moment. Yeah, so you see on the conditions, yeah, and you can directly go from the plate basically to the grid and take a look on the different preparations. And in this case, for example, yes, so something like glycerol cubipol was able even to give directly high-resolution pictures and basically a structure. And this magic works, as I said, with any other protein classes. So with our platform, as I said in the beginning, yeah, so actually everything started as we started to make our membrane protein catalog, and we just recognized that when you do this by the hand in the lab, yeah, so it's just taking way much time, yeah. And the question was how we can reduce this time. And we started with our membrane catalog and started to search basically for very different targets that we think are interesting for our customers. And now we have a portfolio with more than 350 proteins that are available as off-the-shelf production, so to say, so it will be produced especially for you in this moment. And this is our LinkedIn story, basically. Every Wednesday, that's already since 2 years tradition. Yeah, so our service team, I'm part of the service, and my colleagues, we're posting every week a new target. And you see here basically always the results of the screening. You see very different proteins, yeah, so which are basically available, but there is one common thing. They come very pure, directly from the first affinity purification, and the DLS profiles are remarkably clean, yeah, so, and showing a very good particle distribution. So giving you a bit overview of what we have. So this would be different protein classes that we have. Maybe you see already the target that you're working with, or you struggle to work with, better say. So they are already available. So that includes ABC transporters, occoporins, claudins, diverse GPCR classes, integrins, ion channels. And here, the second page, so very different receptors, SLC transporters, tetraspanins, and very, very other proteins. And as I said, the cool thing about this, yeah, so if you even want to study a bit more, you know, the biology of the protein in more detail, yeah, so the great feature of synthetic copolymer is that if any protein has an interaction partner in the membrane, we can basically co-extract it. The main important thing is for this, that the interaction is tight enough so that they're close enough for each other. So basically summing it a bit up, the great achievement so that we have with this plate is that simple as it is. You have your cell lysate, you establish your expression, you take your cell lysate, you take the plate XMP from Biotech, you put it in the liquid handler, and in 2 hours you get the LOAs of your membrane protein of interest. If you have, just in case, if you have some kind of very struggling target, yeah, so, and this solution of just only like 8 for example, screening condition is not enough, what we also offer is this custom preparation, more or less. Yeah, so we can go on combination on something we call NativeMP plate and our pure HT plates, where we can basically put way more copolymers inside of the plate, which would be custom for your target, for example, in this case. So you can do then 2, 4, 6, or even 16 preselected copolymer on each plate, and then you need the combination basically of plate with the synthetic copolymers and plate with magnetic beads, for example, as I said, with very different flavors in this case, and then we would do a kind of a custom plate for you so that you can screen way more conditions, for example, on how to, if you would like to optimize the process, for example, as too. So our take-home message, more or less, would be today the membrane. We think in Kiev Biotech, so that's this kind of the stabilization is the future of the membrane protein research. It doesn't necessarily mean that detergents are bad and you don't need to, we cannot trust the results or something like this. It's also a way to work with this, but we want to improve. We want to have the results that we can trust, and we want to work as native as possible. So, and we're showing every week, every day that the NativeMP system is working well. So with this, thank you very much for coming. If you would like to speak more or you have any additional questions that we discuss in depth, we are at the booth 700 in the exhibition hall. Also, don't hesitate to visit the colleagues from Analytic Jena, so if you want to learn more about their automatization solutions. And thank you very much for your attention, and I'm happy to receive the questions if you have any. Thank you. Okay. Do you have any questions, please?
I wanted just to ask whether you have any experience in using protein particles for immunization or animal immunization.
Yes, so usually, so what I mentioned also in the downstream applications, so we have a case study where it was done in vitro, so with the phage display, but in service we do oftenly have customers who immunize animals, so it mouses, mice, rats, rabbits, alpacas, and so on. Yeah, so it's of course some data I cannot disclose in the end. Yeah, so, but it's a well-known application, so to say, also with these particles, because mostly, you know, when you, for example, have a detergent preparation, yeah, once you inject it to the animal, yeah, so of course the level of the detergent drops, yeah, so you cannot be sure that the protein is still there, yeah, so what people also usually do is to extract this extracellular domains, for example, something what I've shown, yeah, so but we always, or use peptides, for example, as well, yeah, so but we always ask, like, how the antibodies are done in the end, because when you then go on cellular assays, so sometimes you see a problem with the bindings, yeah, so because the conformation is compromised in this moment, yeah, so and this is something we always tell our customers from coming from the antibody, nanobody discovery, for example, that basically try to be in this moment as native as possible, that minimizes your risks in terms of the quality of the antibody in the end. Thank you. Any more questions? Yeah, then thank you very much for coming. And as I said, at the booth 700, please go ahead. We have beautiful bags with a giveaway, yeah, so with our beautiful small animals, like not real ones, yeah, so, but they are pretty cute, yeah, so thank you very much.